Chapter 7 of your Molecular Diagnostics textbook will be covering amplification of nucleic acids. Early analysis of nucleic acids were limited by the availability of material for analysis. Generation of enough copies of a single gene required propagation of millions of cells in culture, or isolation of a large amount of genomic DNA. The ability to amplify a specific DNA sequence opened the possibility of analyzing at the nucleotide level virtually any piece of DNA in nature. In this chapter, we'll be describing the principle of amplification by polymerase chain reaction, also known as PCR. We'll be discussing how products are detected, describing examples of modifications that have been developed for PCR, differentiating between target amplification and signal amplification, and comparing and contrasting between the following in vitro assays for amplifying nucleic acids: PCR, branched DNA, ligase chain reaction, transcription-mediated amplification, and hybrid capture. The first specific amplification method of any type was polymerase chain reaction. It involves target amplification. Target amplification can make copy numbers in the millions until the sequence can be detected. This process is similar to growing cells in culture, in which the cells are allowed to replicate their nucleic acid as well as themselves so that they can be visualized on an agar plate. Culture in vivo can take days, whereas in vitro replication takes only hours. The process of in vivo replication involves double-stranded DNA that serves as a template, DNA polymerase to catalyze addition of nucleotides to the growing strand, and primer, which is synthesized by primase in vivo, to which DNA polymerase adds subsequent bases. Polymerase chain reaction duplicates in vivo replication of DNA in vitro. It uses primer-directed in vitro enzymatic reaction for the production of a specific DNA fragment. Figure 7.1 in your textbook shows the components and results of PCR. Oligodeoxynucleotides, or primers, are designed to hybridize to sequences flanking the DNA region being analyzed. The polymerase extends the forward and reverse primers, making many copies of the region, flanked by the primer sequences, the PCR product. The DNA is replicated with the same end result, one copy of double-stranded DNA becoming two copies. The components of PCR include a DNA template, short oligonucleotide primers, nucleotides, polymerase, and buffers, and these are all subjected to an amplification program, which consists of a specified number of cycles that are divided into steps, during which the samples are held at particular temperatures for designated times. Primers are critical components of PCR because they determine the specificity of polymerase chain reaction, similar to the probes we saw in blotting and hybridization procedures discussed in Chapter 6. The design of the primers require knowledge of the target sequence, and the placement of the primers will dictate the size of the amplified product. Primers are single-stranded 18 to 30 base DNA fragments. They're complementary to the flanking region to be amplified to ensure replication occurs at the exact region of interest and includes the entire section. This slide gives a high-level overview of a PCR procedure. When performing PCR, a reaction mix needs to be assembled containing all the components necessary for DNA synthesis. This includes some of the PCR components we discussed earlier, such as the short oligonucleotide primers, nucleotides, or polymerase. The next step is subjecting the reaction mix to an amplification program, and then the last step is analyzing the product of the PCR reaction. When the PCR procedure is complete, millions of copies can be produced, and these are called amplicons. Table 7.1 in your textbook provides a typical PCR reaction mix. So the 0.25 millimoles of primer are used to direct DNA synthesis to the desired region. The 0.2 millimoles of DATP, DCTP, DGTP, and DTTP are the building blocks that extend the primers. The 50 millimoles of potassium chloride are added as a monovalent cation or a salt for optimal hybridization of primers to the template. The 10 millimoles of tris at a pH of 8.4 are required to maintain optimal pH for the enzyme reaction, and this is a buffer. The 1.5 millimoles of magnesium chloride are a divalent cation that are required by the enzyme. The 2.5 units of polymerase are the enzyme that extends the primer. And finally, the 10 to the second to 10 to the fifth copies of the template are the actual sample DNA that's being tested. When performing PCR, amplification takes place as the reaction mix is subjected to an amplification program. The amplification program can consist of a series of anywhere from 20 to 50 PCR cycles. The PCR cycles are a three-step process. It starts with denaturation at 94 to 96 degrees Celsius for 20 seconds. The next step or cycle is annealing at 50 to 70 degrees Celsius for 20 seconds. And the third step is the extension step at 68 to 72 degrees Celsius for 30 seconds. And we'll discuss these in a little more detail on the subsequent slides. In the first step, the double-stranded DNA is denatured into two single strands. This is accomplished by heating the sample at 94 to 96 degrees Celsius for several seconds to several minutes, depending on the template. The initial denaturation step is lengthened for genomic or other large DNA template fragments. Subsequent denaturations can be shorter for these larger fragment types. Step 2 of the PCR process is the most critical step, and this is annealing. The two oligonucleotide primers that will prime the synthesis of DNA anneal or hybridize to complementary sequences on the template. In other words, the primers determine the specificity of the amplification. It's very important that the annealing temperature be optimized with the primers and reaction conditions. The temperatures usually range from 50 to 70 degrees Celsius. Of note, a concept discussed in Chapter 6 regarding stringency and hybridization involves reaction conditions, salt concentration, mismatches, template conditions, all affecting the melting point of the primers in the reaction. The final step in PCR in which DNA synthesis occurs is step 3, known as extension. The polymerase synthesizes a copy of the template DNA by adding nucleotides to the hybridized primers. DNA polymerase catalyzes the formation of the phosphodiester bond between an incoming dNTP and replicates the template. Remember, this is done in an antiparallel and complementary fashion, so the replicated piece is not identical. Optimal temperatures for this step are 68 to 72 degrees Celsius. And at the end of these three steps, one cycle has completed and one copy of double-stranded DNA has been replicated into two copies. Returning to the denaturing temperature of 94 to 96 degrees Celsius starts the second cycle, with the end result being a doubling in the number of double-stranded DNA. So after the second cycle, 4 double-stranded DNA are produced. By the third cycle of PCR, 8 double-stranded DNA are produced, so the copies increase exponentially. The PCR product, as mentioned before, is an amplicon, and the number of amplicons is 2 to the nth power, where n is the number of cycles of PCR. So if we continue cycling from the previous slide, the fourth cycle would equal 16 copies. The fifth cycle would equal 32 copies. The sixth cycle would equal 64 copies, and then they would continue on to the nth power, depending on how many cycles in the PCR procedure being done. The first PCRs were performed using multiple water baths or heat blocks set at the required temperatures for each of the steps of the PCR cycle. The tubes were manually moved from one temperature to another. In addition, before the discovery of thermal stable enzymes, new enzymes had to be added after each denaturation step, further slowing the procedure and increasing the chance of error and contamination. Automation of the tedious process was greatly facilitated by the availability of heat-stable enzymes. Thermal cyclers change the temperature in a block or chamber that holds the samples. And the thermal stable polymerases are used to withstand the repeated high denaturation temperatures. This is an example of internals of an automated PCR instrument that has different components to facilitate the temperature changes and to analyze multiple samples. As you can see in this diagram, there's several different areas where there's chamber vents and fans to facilitate the movement of air and heating and cooling. As mentioned earlier, automation of PCR was pushed along by the discovery of the thermal-stable enzyme Taq polymerase. DNA polymerase was originally isolated from E. coli, but high temperature inactivated the enzyme, and new polymerase needed to be added to the tube after each round of denaturing. With the discovery of Thermus aquaticus and subsequent use of Taq polymerase, enzyme only needed to be added at the start of the procedure and would maintain its activity throughout. Over time, other enzymes were discovered that had utility in variations of the PCR method. After PCR, a variety of methods are used to analyze the product. Most commonly, the PCR product is analyzed by gel or capillary electrophoresis. Depending on the application, the size, presence, or intensity of PCR products is observed after electrophoresis. Things to keep in mind when performing testing and interpreting results are to rule out any contamination. So the reagent blank should be negative and controls should be appropriately amplified. The PCR product should be of the expected size. It should be kept in mind that misprimes may occur due to nonspecific hybridization of primers, and primer dimers may occur due to hybridization of primers to each other. This is an example of PCR products after resolution on agarose gel and staining with ethidium bromide. Molecular weight markers in the first lane are used to estimate the size of the PCR product. The intended product in this example is 125 base pairs. Primer dimers and misprimed product are also present in this example. So here we have misprimed product, and here are primer dimers. The absence of product in the last gel lane, labeled the reagent blank, confirms that no contamination occurred in the master mix. The master mix is the reagent and buffer combined that can be pre-prepared when performing PCR procedures. Once ready to perform the PCR procedure, the enzyme, target, and primers can be added to the master mix when ready to start the procedure. The next few slides discuss methods to ensure that the PCR product is adequate and ways to control possible misprimes or contamination. So in order to avoid misprimes, proper annealing temperatures need to be used, and the primers need to be designed carefully. Adjustment of the monovalent cation concentration can also be done. A hot start method can be used in which reaction mixes are prepared on ice and then placed in the preheated cycler, or use of a sequestered enzyme that requires an initial heat activation can be used, such as platinum Taq, AmpliTaq, gold, or hot star Taq. Primer design is a critical aspect of PCR. It's important to avoid using primers that may anneal to the incorrect sequence due to similarities in target. Hybridization of primers is subject to the same physical limitations as probe hybridization. The primer sequence, or the percent of GC content, and length affect the conditions in which the primer will bind to its target. The approximate melting temperature, or TM, of the primers can be calculated calculated using the same equations for short DNA fragments that were described in Chapter 6. This primer then serves as a starting point for setting the optimal annealing temperature, the critical step for the specificity of amplification reactions. Primers should be designed such that the forward and reverse primers have similar melting points, so both will hybridize optimally at the same annealing temperature. Melting point or melting temperature can be adjusted by increasing the length of the primers or by placing the primers in areas with more or fewer G's and C's in the template. The GC content of the primer should be between 20 to 80%. The product size should be 100 to 700 base pairs. And the primer should be as specific as possible to the target of interest. Limitations in primer design or reaction conditions may not completely prevent primer dimers or other extraneous products. Unintended amplicons may make the product unacceptable for some analytic procedures such as sequencing or mutation analysis that require pure end products. Residual components of the reaction mix, such as leftover primers and unused nucleotides, may also interfere with some post-PCR applications. The choice of cleanup procedure or whether cleanup is necessary is dependent on the application to be used. Gel elution removes all reaction components as well as misprimes and primer dimers. Solid phase isolation of PCR products such as spin columns bind DNA to a column and rinse away the rest of the reaction components by centrifugation. DNA precipitation is a method used to purify or concentrate DNA by adding compounds such as ethanol. When considering contamination control in the PCR procedure, any molecule of DNA that contains the intended target sequence is a potential source of contamination, and the most dangerous contaminant is the PCR product from a previous reaction. This is why laboratories are designed to prevent exposure of pre-PCR reagents and materials to post-PCR contaminants, and this is why the unidirectional flow is an important concept in PCR workup and procedures. Several different methods can be employed to control contamination. So one of the first is physical separation. So airlocks and positive airflow can be employed along with PCR hoods that utilize ultraviolet light. DUTP plus uracil and glycosylase can be added to the PCR reaction. This is a frequently used method that involves DUTP added to the master mix, resulting in incorporation of DUTP instead of DTTP in the PCR product, because DUTP does not affect the PCR product in most reactions and helps eliminate carryover of previously amplified products. Psoralen and UV can be used. Psoralen is a compound that increases the effectiveness of UV light. Also, another method that's commonly used in every area of the lab is using a 10% bleach. So this is the most effective method for surface decontamination. Polymerase chain reaction can be applied for various purposes in the clinical laboratory. It can be used for structural analysis of genes, DNA typing, disease detection, cloning, analyzing mutations, detection of gene expression, mapping, site-directed mutagenesis, and gene sequencing. On the next few slides, we'll discuss the different modifications of PCR and how those modifications are applied to specific nucleic acid detection. The original method of polymerase chain reaction has been adapted for various applications, and several are used in the medical diagnostic laboratory. Modifications include those such as nested PCR, multiplex PCR, tailed primers, sequence-specific PCR, reverse transcriptase PCR, long-range PCR, whole genome amplification, rapid PCR, and quantitative real-time PCR. Some methods are capable of detecting multiple targets in a single run, such as multiplex PCR. We're going to go into more detail in quantitative real-time PCR or qPCR, since this is one that's more commonly used in the clinical laboratory. Along with the adaptations of the original PCR method, automated methods have been developed as discussed a little earlier. The cobas Amplicon analyzer is another example of an automated instrument. Samples are amplified and products detected automatically after the PCR reaction. The cobas analyzer can be used for several infectious disease applications, including detection of HIV, hepatitis C virus, hepatitis B virus, CMV, chlamydia, Neisseria, and Mycobacterium tuberculosis. Real-time or quantitative PCR, also known as qPCR, is often used in these applications, and products are detected by fluorescence during the PCR reaction. Standard PCR procedures will indicate if a particular target sequence is present in a clinical sample. For some situations, though, the clinician is also interested in how much of the target sequence is present. There are several approaches to estimating the amount of starting template through PCR. Quantitative PCR was originally performed by adding ethidium bromide to regular PCR. The ethidium bromide intercalates into double-stranded DNA and fluoresces, and it can be used to monitor the accumulation of PCR products during the PCR in real time. The detection of a signal in real time allows for quantification of the starting material. Real-time or quantitative PCR is performed in a specialized thermal cycler with fluorescent detection systems. With quantitative PCR, the product grows in an exponential fashion. It doubles at each cycle as seen with the standard PCR. The PCR signal is observed as an exponential curve with a lag phase, a log phase, a linear phase, and a stationary phase. The lag phase is the initiation as the reaction components are in excess and copies begin to be made. The log phase is when copies increase exponentially. The linear phase is when one or more components of PCR have decreased below a critical concentration, therefore amplification begins to decrease. And the stationary phase is the endpoint of the reaction. The length of the lag phase is inversely proportional to the amount of starting material. The advantage of this method over standard PCR is its ability to determine the amount of starting template accurately. With quantitative PCR, a threshold level of fluorescence is determined based on signal and background. Plotting the target copy number of the diluted standard against the threshold cycle for each standard generates a graph in which the relationship is established, and then the starting amount of unknown specimen can be determined by the cycle number at which the unknown crosses the fluorescence threshold. The figure on the left shows a plot of the accumulation of PCR product over 50 cycles of PCR. This is a sigmoid curve. The generation of fluorescence occurs earlier with more starting template than with less. On the right, the cycle number at which fluorescence increases over a set amount or the fluorescence threshold is inversely proportional to the amount of starting material. Detection systems utilized in quantitative PCR include DNA-specific dyes, hybridization probes, and primer-incorporated probes. DNA-specific dyes are used to bind and fluoresce as targets are amplified. Ethidium bromide has been used in the past as it is specific for double-stranded DNA, but it's been replaced by SyBrGreen due to concerns over toxicity. SyBrGreen is also advantageous because it's more specific and has more robust fluorescence than ethidium bromide. The use of probes to generate fluorescence have also been employed as a means of detection of DNA. Hybridization probes such as TaqMan have been developed that use the natural 5' to 3' exonuclease activity of Taq polymerase to generate a signal. Molecular beacons are another probe-based detection system that measures the accumulation of a product at the annealing step in the PCR cycle. Dyes such as ethidium bromide and SyBrGreen are still used for routine quantitative PCR, but the use of nonspecific dyes can also be used. These require clean PCR with no mispriming or primer dimers because those artifacts will also generate fluorescence, giving a nonspecific result. Hybridization probes are more specific and only bind and fluoresce the intended PCR product, and primer-incorporated probes label the PCR product. The next few slides go into the different detection systems in a little more detail. With non-sequence specific dyes such as ethidium bromide and SyBrGreen, binding of double-stranded DNA products of PCR occurs, and as more copies of the target sequence accumulate, the fluorescence increases. As the fluorescence increases with product accumulation, a melt curve can be graphed in which specific sequences have characteristic melting temperatures. Once this relationship is established, the starting amount of an unknown specimen can be determined. This figure shows a diagram of TaqMan quantitative PCR. In real-time quantitative PCR using TaqMan as the detection system, the probe hybridizes the target sequences between the primer binding sites. The probe is covalently attached to a fluorescent reporter dye at the 5' end and a quencher at the 3' end. TaqMan signal fluorescence is generated when Taq polymerase extends the primers and digests the probe and releases the reporter from the vicinity of the quencher. Another quantitative PCR detection system is a molecular beacon probe. These type of probes contain target-specific sequences and a short inverted repeat that hybridizes into a hairpin structure. The 5' end of the probe has a reporter dye, and the 3' end has a quencher dye. As mentioned earlier, they measure the accumulation of product at the annealing step in the PCR cycle. Molecular beacons bound to the target sequences fluoresce, and fluorescence doubles with every doubling of target sequences. FRET probes can also be used as quantitative PCR detection systems. FRET stands for Fluorescent Resonance Energy Transfer, and it utilizes two specific probes. One with a 3' fluorophore, known as an acceptor, and the other with a 5' catalyst for fluorescence, the donor, that bind to adjacent targets. The acceptor-reporter complex will fluoresce only when both probes are bound next to one another on the target sequences. As more target accumulates, more probe binds and more fluorescence is emitted. FRET probes are used in the medical laboratory for viral detection and quantitation, and for amplification and detection of genetic diseases. As we saw earlier, automated detection systems include equipment such as thermal cyclers that rapidly and automatically change the required incubation temperatures, holding each for designated periods. These systems include fluorescent detection and specialized software to detect and analyze products and produce results. With these systems, PCR reactions take place in optically clear plates, tubes, or capillaries, such as the Cepheid Smart Cycler clear plate or the Roche Light Cycler capillary tubes. There are many advantages to using PCR in the clinical laboratory. With advances in technology, it has become a simple, rapid, and relatively inexpensive test that is specific for the target of interest. It has numerous applications, such as detecting bacteria or viruses that cause infectious diseases, or gene mutations that cause genetic defects. PCR is sensitive enough to detect low quantities of nucleic acids and can even be utilized if DNA is damaged. It has become increasingly user-friendly and more automated in the past several years, and has numerous applications in the various laboratory specialties. Although PCR has many advantages, there are also limitations to the method. Risk of contamination is always a limitation, and it can be either pre-analytical or analytical, depending on how the specimens are collected and how testing is performed. For example, if less automated procedures are utilized, it increases the risk of contamination. Other limitations are the availability of primers for more complex or less frequently encountered target sequences, along with the complexities that may occur with primer binding sites. The possibility of amplification of rare targets is also a limitation that may interfere with accurate results. And finally, the various detection methods cannot be used for all applications, and careful consideration must be taken when deciding which detection method should be used in respect to the target sequences that are being analyzed. Aside from PCR, other amplification methods can be utilized, and we'll discuss these on the next slides. These include probe amplification and signal amplification methods. The various applications include ligase chain reaction, branched DNA, hybrid capture, and transcription-mediated amplification. Ligase chain reaction is a probe amplification. This type of amplification involves synthetic probes that are specific to the target sequences that bind to the target, and the probes themselves are amplified instead of the target nucleic acid. In this method, copies of target molecules do not change. It's the probe that's amplified. Similar to PCR, ligase chain reaction requires a thermal cycler to change the temperature to drive the different reactions. In ligase chain reaction, the probes bind immediately adjacent to one another on the template. The bound probes are ligated and become templates for the binding of more probes. This type of amplification method is used in detecting chlamydia trachomatis and Neisseria gonorrhea, along with sickle cell mutations. In this diagram, you can see that the ligase chain reaction generates a signal by repeated ligation of probes complementary to specific sequences in the test DNA. One complementary oligomer is covalently attached to biotin for immobilization. On the diagram, this is the yellow square. And one has a signal-producing molecule, which is depicted as the purple circle on the diagram. The two oligomers will be ligated together only if the sequence of the target is complementary. The oligomers captured on a solid substrate by streptavidin will generate a signal. If the sequence of the target is not complementary, the capture probe will not yield a signal. Ligase chain reaction, discussed on previous slides, is a probe amplification method. Branched DNA is a signal amplification procedure. Signal amplification involves no change in the number of target or probe sequences, but instead large amount of signal are bound to the target sequences that are present in the sample. Signal amplification procedures are inherently better at quantifying the amount of target sequences present in the clinical sample. Branched DNA involves a series of hybridizations that attach multiple signals to each target molecule. This amplification method has been used for hepatitis B virus, hepatitis C virus, and HIV-1. In branched DNA amplification, a series of short oligomer probes are used to capture the target nucleic acid. Additional extender probes bind to the target nucleic acid and then bind to multiple reporter molecules, loading the target nucleic acid with signal. The target is captured or immobilized to a solid support by capture probes, after which extender probes create a stable cruciform structure with amplifiers. Each amplifier has hybridization sites for 8 to 14 branches, which in turn bind substrate molecules for alkaline phosphatase labeled oligonucleotides. Chemiluminescence after these reactions takes place is measured in a luminometer. Hybrid capture is another signal amplification technique in which the target DNA is not amplified. Instead, the DNA is isolated, bound to RNA, and is recognized by multiple antibodies to the target probe hybrid molecule. This assay has been marketed primarily for detection of human papillomavirus in genitourinary specimens, but is also available for detection of hepatitis B virus and CMV. Hybrid capture starts with hybridization of the RNA probe to the denatured DNA target. The RNA-DNA hybrid is then bound by hybrid-specific immobilized antibodies. A secondary antibody bound to alkaline phosphatase generates signal in the presence of a chemiluminescent substrate. The chemiluminescence is subsequently measured to determine the presence of the target. The last type of amplification method we'll discuss is transcription-mediated amplification, or TMA. This is a transcription-based amplification system in which RNA is the target instead of DNA. It differs from other nucleic acid amplification procedures in that RNA is both the target and the primary product. A DNA copy is synthesized from the target RNA, and then transcription of the DNA produces millions of copies of RNA. Several commercial variations of this process are available, and they may be called TMA or NASBA, nucleic acid sequence-based amplification, or 3SR, self-sustaining sequence replication. The names of the method vary depending on the manufacturer of the assay. Transcription-mediated amplification has some advantages over PCR and other amplification procedures. It's an isothermal process, negating the requirement for thermal cycling to drive reactions. Another advantage is that it targets RNA, allowing for direct detection of RNA viruses, and can also target RNA of organisms with DNA genomes, which is more sensitive than DNA. This is advantageous when detecting RNA from bacteria that have multiple copies of RNA, but only one copy of DNA. In transcription-mediated amplification, complementary DNA is made from the RNA target, adding RNA polymerase promoter. RNA is synthesized from the complementary DNA template and can serve as a source of new complementary DNA. Transcription-mediated amplification methods can be used to detect Mycobacterium tuberculosis, Chlamydia trachomatis, HIV, and CMV. The first step in transcription-based amplification is the production of a complementary double-stranded DNA copy of the RNA target. Synthesis is performed by reverse transcriptase, which extends a primer that is tailed with an RNA polymerase binding site sequence. The RNA-DNA hybrid is digested with RNase H, leaving the single-stranded DNA, which is converted to a double strand with a complementary primer. The complementary DNA produced in the first step serves as a template for RNA polymerase. Many copies of RNA are subsequently synthesized, which are primed by a complementary primer for synthesis of another RNA-DNA hybrid. Target-specific probes are then bound to the RNA, and the target amplicon can be detected. So this concludes Chapter 7, Nucleic Acid Amplification. In summary, PCR is a method to specifically amplify target sequences in a complex mixture. The primers determine what sequence are amplified. Contamination control is important in laboratories performing PCR. Quantitative PCR offers the advantage of quantifying target. And in addition to PCR, signal and probe amplification methods are available for use in the clinical laboratory.